Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221. This is lecture number 9 of our series of our online lectures. The topic of our today's lecture is post-transcriptional modifications. As you studied in the process of transcription, we get RNA, but this RNA is processed before it is used for different purposes. So we study about those processes in post-transcriptional modification. The outline for today's lecture is we will talk about introduction to post-transcriptional modifications, and in this we will study what these post-transcriptional modifications are. We will study about messenger RNA processing. Then we will study about messenger RNA splicing. We will talk about polyadenylation. This is the process in which poly A tail is added to the messenger RNA during post-transcriptional modification. Then we will study about transfer RNA and how this transfer RNA is processed. We will study about processing of ribosomal RNA or rRNA. So this is the outline of today's lecture. Now we move towards our lecture. Post-transcriptional modification or RNA processing, which is also alternatively named as RNA processing. It is a process by which primary transcript RNA is converted into mature RNA. Though it is critical to eukaryotic messenger RNA, it is responsible for changes in rRNA, tRNA, and other special function RNAs too. So overall, it covers the different types of RNA that we have, mainly including messenger RNA, transfer RNA, ribosomal RNA, and other special function RNAs too. Now, we see that here there are two types of broad categories of organisms that we have. One are prokaryotes, and in prokaryotes, this is how a prokaryotic cell looks like. The DNA is here. Messenger RNA in the red, you can see here, it is when it is formed, when the process of transcription occurs, we see that these are ribosomes, the yellow, the two subunits, nascent protein are formed. So this is generally coupled transcription and translation. The processes of transcription and translation are coupled. It means that when the RNA transcript is formed, it is at the same time the ribosomal machinery converts these to translate it into proteins. One can see by this blue the nascent protein formation. However, in eukaryotes we have DNA that is inside the nucleus. You can see here the nucleus. This is the DNA, and this is in the red you can see the primary transcript of RNA. So now this RNA is in the red, you can see here it is from 5' to 3'. If this is messenger RNA, it is transported out of the nucleus to the cytosol. Here in the yellow you can see the ribosome, and when it is translated, the blue color are showing the nascent protein, the proteins that are formed. So messenger RNA is processed and transported out of nucleus for translation. This is in case of eukaryotes. While in prokaryotes, one can see the coupled transcription and translation processes. So now, we know that from DNA, through the process of transcription, we get the primary transcript. Now this primary transcript is processed to tRNA or transfer RNA. It is also processed to messenger RNA or mRNA, and it is also processed to rRNA or ribosomal RNA. So these are the three major matured RNAs that we have. We get as a result of a post-transcriptional modification of the primary transcript. Now, this process of processing occur in different steps. Now we focus on messenger RNA processing. Messenger RNA processing require different steps. One of those steps is capping, that is addition of 5' to 7-methyl guanosine cap, and then we get splicing to remove intervening sequences or the introns, and then polyadenylation or addition of a poly A tail at the 3' site takes place. Now, this is the primary transcript of a messenger RNA. This is start site of RNA synthesis, and this is where the poly A tail at the 3' end is added. So now to look at the more simplified version of this is primary RNA transcript having 5' end on this side, and the 3' end is on this side. Now 3' cleavage and addition of poly A tail occur. You see here multiple poly A are added. N shows the number of poly A that are added, so a poly A tail is added. Now we have the red color showing the exons. The blue ones are the intron. You can see it here, and this gray color is showing the UTR. So in the next step, you can see the blue, the introns, they are removed, they are excised, and intron excision and exons are ligated with each other. So at the end of the process, we get the exon ligated together. So this becomes a mature RNA or mRNA. So how this happens? Now we talk about this. Now 5' capping. You see here these three processes: capping, splicing, and polyadenylation. We talk about the detail of these individual processes. This paragraph overall is showing the overall process, but now we go in more details and see how all this is happening. So 5' end of RNA with triphosphate group. The alpha, beta, and gamma phosphates are shown here. This is the 5' end showing nucleotide with these alpha, beta, and gamma phosphates. Phosphohydrolase enzyme will remove one of the phosphate, and now we are left with this nucleotide having 2 phosphates. Now after this, with the help of guanylyl transferase, this nucleotide, this is 7-methyl guanylate, this cell will come here. The 2 PPIs will be removed, and the phosphate is this group is attached here. You can see now, along with one of its phosphate. So after this, adomate will enter the system. Guanine 7-methyl transferase will function here, and it will the ADO-HY, it will be removed. It will result in formation of this. Now again adomate and will enter another one. Will enter and this ADO-HY, this will be removed. This will be done with the help of 2'-O-methyl transferase, and as a result of this, we get 5' end of RNA with K. So this is showing how the 5' linkage is happening. So this slide tells us how the 5' capping of the RNA takes place. Now this cap have different functions. This cap provides protection from some ribonucleases degradation, because this RNA has to be transported from the nucleus to the cytosol, and the environment is rich with ribonucleases. So this cap will help and provide protection from ribonucleases, so it will not be degraded. It further stabilizes the messenger RNA. It enhances translation and splicing. And it also functions as enhanced transport from nucleus to cytoplasm. So the capping provides different essential and protective role in stabilization and helps in translation and transport of the RNA. Now, there is another process which is called messenger RNA splicing. Messenger RNA is called hnRNA, or heterogeneous nuclear RNA, before splicing occurs. Now, this heterogeneous nuclear RNA proteins to help keep the hnRNA in a single-stranded form and to assist in the various RNA processing reactions. Exon and intron lengths and numbers vary in various genes. So they're not fixed. It depends on the gene, the type of gene that is being transcribed. So now, what is exon? Expressed sequences is any segment of an interrupted gene that is represented in the mature RNA product. Intron, intervening sequences, is a segment of DNA that is transcribed but removed from within the transcript by splicing together the sequences, exons on either side of it. Splice junction consensus sequence. Now you see here, this portion is showing the exon, this is showing the intron, and then again here it is the exon. Now this is called the 5' splice site, and this is called 3' splice site. AG rule describes the presence of these constant dinucleotides at the first two and last two positions of nuclear genes, GU and AG. So splice sites are the sequences immediately surround the exon-intron boundaries. Splicing junctions are recognized only in the correct pairwise combinations. The sequences of steps in the production of mature eukaryotic messenger RNA is shown for the chicken ovalbumin gene. Here you see ovalbumin gene with 7007 base pair. You see here is the one intron, here is the exon. Intron, exon, intron, exon. So it consists of this primary transcript, which is 7700 base pair, consists of introns and exons. Now, after transcription, we get the primary transcript, which is the hnRNA. We get this primary transcript. Now, capping and polyadenylation of this will result in the formation of cape here, and the poly A tail is added here. Now, once this is done, this is followed by splicing. Splicing will remove the introns, the non-coding portion, and it will the exons, the coding portion will be ligated. Ovalbumin messenger RNA, now that is mature, it consists of 1872 nucleotides. It was before this it was consisting of 7700 base pair, but now it is only 1872 base pair because of the removal of the introns through the process of post-transcriptional modification. Now this is the mature messenger RNA that we have for this particular gene. Now, messenger RNA splicing, splicing is mediated by a large ribosomal nucleoproteins complex called a spliceosome. Spliceosome contains a specific set of base uracil-rich SNRNPs or small nuclear RNPs associated with proteins, SNRNA complex with protein. Function of SNRNPs: recognizing the 5' splice site and the branch site, bringing those sites together, catalyzing or helping to catalyze the RNA cleavage. Spliceosome complex: splicing SNRNPs, it consists of these are 5 different units U1 is 5' site recognition here. U2 function is branch site recognition, you can see here. U4 forms base paired complex and acts with U6 here, so U6. U5 3' junction binding of U4 U6 complex. U6 complex with U4 makes spliceosome transesterase. Spliceosome recognize introns starting with 5' GU and ending in AG 3'. Now, mechanism of spliceosome. Here you can see exon 1 GU splice site. Here is the branch site. Here is exon 2 AG 3' splice site. U1 binds to 5' splice site, U2 binds to branch site. You see, U1 binds to 5' site, U2 binds to branch site. U4 and U6 and U5 trimer binds intron, loops out, and exons are brought closer together. You see, exons are brought closer together. Intron loops out and exon brought closer together. You can see here. Now, splice site is cut. 5' splice site is cut. 5' end of intron is connected to the A in the branch site to form a lariat. U1 and U4 are released. 3' splice site is cut. Exon 1 is connected to exon 2. The intron in the form of a lariat is released along with U2, U5 and U6. Intron will be degraded because they are non-coding sequences which are not needed. Intron plus U2, U5 and U6 here. Intron will be degraded in the SNRP is used again. So here exon 1 and in exon 2, two connected exons are shown here in the red. So you see the intron removed with the help of these different subunits. Now there is another way, and that is called alternative splicing. Pre-mRNA are spliced in several different ways, allowing a single gene to code for multiple proteins. The generation of different mature mRNAs from a particular type of gene transcript can occur by varying the use of 5' and 3' splice sites. So you see 1, 2 and 3 exon. These are introns. So splicing can result in either joining of 1, 3, or it can possibly connect 1, 2 and 3. So this is how sex determination in Drosophila takes place, but this is showing alternative splicing, that it can help allowing a single gene to code for multiple proteins with the help of alternative splicing. Polyadenylation, or you see here, this is the polyadenylation complex. 10 to 30 nucleotides are here. Less than or equal to 30 nucleotides are here. GUH or URH. Cleavage takes place. So GUH or URH degraded in the nucleus. Poly A addition takes place, is approximately 250. Consensus sequence for 3' process. So this is how the polyadenylation takes place. Polyadenylation of messenger RNA is a 3' end. This slide overall is showing the polyadenylation process, how it takes place. CPSF cleavage and polyadenylation specificity factor binds upstream, signal 5' end. CSTF cleavage. So this is a detailed process, quite self-explanatory. I would request you to have a look at this, and you will understand how this polyadenylation process or polyA-tail is rated, because if I'm going to describe it, will increase the video length, which will become difficult to upload. So hopefully you will, after reading this, you will be able to understand it yourself how this process of polydenylation takes place. But the ultimate aim of polydenylation is to aid this poly A tail, which will be important for the maturation or the processing of the primary transcript of RNA to aid this. Now, matured mRNA, we get this cap, start site, the coding sequence stop, and then UTR, and then the poly A tail, 5' cap, 5 untranslated region, start codon, coding sequence, stop codon, and 3 untranslated region, poly A tail. You see, this is how a matured messenger RNA look like. Then we have transfer RNA, soluble RNA, supernatant RNA, adapter RNA. Smallest among RNAs, 75 to 93 nucleotides, recognizes codon on messenger RNA, shows high affinity to amino acids, carry amino acids to the site of protein synthesis. Transfer RNA is transcribed by RNA polymerase 3. tRNA genes also occur in repeated copies through the genome and may contain introns. So this is how they recognizes the codon and the anticodon. This is how the transfer RNA looks like. This processing of this transfer RNA occur by removal of leader sequence and trailer, replacement of nucleotide, modification of certain bases. The details of this whole process is given here in the sliding. If you read it, you will be able to understand how the processing of tRNA takes place. tRNA processing and maturation, here is another view. This is a ribozyme, RNA's pCart. This is the primary transcript. The cleavage, addition, different modifications occur in splicing, resulting overall maturation of this tRNA. So RNA can act as an enzyme and catalyze reaction, including its own replication. That is called ribozyme. So this is how tRNA processing. And now finally ribosomal RNA or rRNA in cell greater than 80% of rRNA serves to release messenger RNA from DNA. It is ribozymes and protein synthesis, relatively GC-rich. Ribosome prokaryotes have 70S or 50S and 30 subunits. Eukaryotes have 80S, 60S and 40S subunits. Prokaryotes, 50S subunit is 23S and 5S, and 30 subunit is 16S. So eukaryotes is 60 subunits and 28S, 5.8 and 5S, and 40 subunit is 18S. So these are the different proteins which make up this processing of the ribosomal RNA. Processing of 45S molecules occurs inside nucleus. 45S molecule tightly associated protein forming RNPs. First cleavage occur at site 1 and remove 5' terminal data sequence. So this is the detail of the how the processing of the ribosomal RNA occur. This is the diagrammatic representation of processing of ribosomal RNA. Transcribed spacer 18S, 5.8S, and transcribed spacer cleavage and the detail. So this is the lecture of the different post-transcriptional modifications of RNA that we have. If you have any question, I will be happy to answer. So thank you very much for watching the video.